Vicky, are you able to hear me?
Hello, Wendy?
Ja, oké, it
Yes.
works.
Oh, ik merk alleen dat hij die van mij niet pakt.
Oh ja, ik kan jou wel horen.
Ja, alleen hij pakt mijn headset niet. Want ik hoor jou via de speaker en niet via...
Even kijken, nu als het goed is wel. Denk ik.
Hi, everybody.
Hello.
Hi, Matt.
Good evening.
Hi, everyone.
Yeah.
Yeah, happy to record the session or something.
Yes, it's already being recorded by me, I think.
Great. Dimitri can't make it. He's unwell.
Okay.
So in fact, we don't really have much of an update for the synthesis stuff actually from over here. That may cut off a little bit of an agenda item.
Yeah.
I just put up a page we can use for the meeting there if we want it. Interestingly, I tried something new just earlier. I created a team and added everyone to it, and then I tagged the whole team in that post I just put up there. Did everybody get an alert or like a email alert to the fact that I'd done that?
Let me check.
It's meant to sort of allow you to, ping everybody who's part of a team. That's the theory.
I didn't. Yeah, I got an email earlier this afternoon, actually, but I don't know if that was the email you were meaning.
Okay.
So that was also displaying this Zoom meeting.
Yeah. It was. So I just wrote that, and then rather than tagging everyone individually, I just tried to tag the team, and I don't know if it worked, but it should have worked. We'll see. Rather than typing everyone's name, it's meant to tag everyone in the whole thing.
Yeah.
At least we're all here at the right time. That's positive.
How is everyone over the holiday, by the way? Just, yes, like Christmas, holidays, New Year.
Seems like a long time ago.
Yeah, we're in lockdown, but not as bad as Matt is, I guess.
No, not as bad.
Well,
We are locked down good and proper.
Yeah.
man, it's been a while since I've done all any chemistry related stuff for mycetoma.
Seems like it.
Wait, wasn't it yesterday Australia Day?
Yes, it was.
Happy Australia Day.
Thank you. And it was also quite a hot day, so I didn't really do much. It was like 40 degrees yesterday, so.
It's snowing where I live now.
I would happily trade this for Australia Day, what we have here at the moment.
40 is very uncomfortable if you haven't tried 40 before. So
I've had 40 while living in Adelaide for a while. Yeah, that was not great, but it's still better than freezing cold.
I much prefer like just snow on me. Like I'm getting like sweat everywhere, and it's don't like it.
Yeah, I think that we prefer this weather also above 40 degrees, actually. We can lit the fire and then we're okay. But with 40 degrees, it's difficult to get the heat out of the house.
Yeah.
That's air conditioned, Wendy.
Yeah, but even then.
Suppose the grass is always greener on the other side. Right. Are we waiting for someone else, or should we just start?
We should just keep going.
Yeah.
Okay. I have a suggestion though. can we, from the Erasmus, start first? Because I would like to show what we have, and then we can sort of talk on what to synthesize next according to what we have found over here.
Awesome.
Is that all right?
Yeah, that sounds really good. Yeah.
Okay, I will try to share my window, share screen. Yeah. Okay, I hope this goes through. Is
Where
that
I am
okay?
currently, it's lovely 3C.
Sorry?
I said regarding whether it was 3C. Sorry, I had my phone was dying.
Okay, is the PowerPoint on and well? Because I can't see.
Oh yes, it's working fine.
Okay, great. So I'm going with the pandemic box first because that requires less energy. So we have tested about 14 compounds in vivo. They are the azoles, benzimidazoles, and MMV compounds, as listed here. We've done in vivo tests, and we have observed that 4 compounds, one from azoles, one from the benzimidazole, and 2 from MMV compounds, they significantly enhance larvae survival, and that's ravuconazole, an azole, benzimidazole, fenbendazole from benzimidazole, and 2 MMV compounds, and the other one is olorofim, and that's the new antifungal. So from this, we've shown that 4 compounds shown increase in larvae survival. So with this data, I went on further to sort of look into the relationships within between log D, area under curve, molecular weight, and survival percentage. Of these 14 compounds are tested in vivo, regardless of if it increased survival or not. So I just put them all in together, look at the relationship, and it's not surprising that I haven't. There's no correlation between these 4: log D, AUC, MV, and survival. It's also because all these 14 compounds or drugs are actually quite different to each other, different mode of action. So it's not a surprise that I didn't actually see any correlation. So I think we discussed it last time that different software gives us different log D values. I mean, we should have done. I didn't calculate the log D myself. I got it from this website, Community for Open Antimicrobial Drug Discovery, and they've tested the pandemic box, all of them against few organisms and causative agents of different fungi and amoeba as well, and they have the log D values there. I don't know what they use to calculate the log Ds, but I just took them anyway. I think if you want to go ahead with the log D, then we have to do it with the software that Ben used to determine the phenarimal analogs. I went further ahead to put in the phenarimal analogs, their log D values and their survival from the paper we published in 2018, these phenarimal analogs. I put them in. They are the triangles, gray triangles. The circles are pandemic box compounds, and the colored circles are the 4 significant, the 4 compounds that increase larvae survival, and 2 compounds that, if you look into the figure, it sort of enhances survival a little bit, but not as much, and that is my mouse here, ebuconazole. As you can see, that the line is quite similar to ravuconazole, except that in the end it drops a little bit. So that's ebuconazole, the gray one here, and luliconazole as well, the orange one. As you can see, the survival was quite high in the beginning, but not so towards the end of day 8 or so. So that as well I've included in this figure, and you can see that, of course, there's no correlation. What we saw from last time was we saw that log D of 2.5 and above, I think it's somewhere here, gives a significant increase in larvae survival, or at least it works well in vivo. That's what we found last time. But I don't think that really we can actually say that here, although we can see that most of the colored circles are above 2.5. I don't think I am in the right.
The outlier that is lower than 2 and lower than 10.
Lower than 2, that's this one.
Yeah, what's that?
I need to double check. I can't really check it here actually, but I'll get back to you with that.
I mean, if somebody put a gun to your head and said, draw a line through that, then you would draw something that indicates lower log D has higher % survival, if you really had to. It's just, it's very scattered.
Yes, yeah, okay, yeah. Let's say about below 5. Okay, but I don't think I'm the right person to look into the what do you call that? Well, let me rephrase that. I think we also need to look into the other aspects of the compounds, not just log D, and that we will need your help with that. We are not that versed with this area.
But I think what you said is
yes.
right. It's very difficult to compare molecules if they have fundamentally different structures and different mechanisms of action. So the original thing we did of doing the log D comparison within the phenarimoles carried more weight because it was within a series.
Yeah, exactly. So I don't think I can say much about this figure and our analysis. But yeah, if you want to look further, we will have to ask for look into other ways of analyzing the data we've got. And actually, we can do that with the phenarimoles. Again, there's 14 phenarimoles tested in vivo, 2 from Dimitri, 3 from the rest from Hong, and 3 from EPL. It's a bit unfortunate, but we've only found 2 compounds that, one compound that increased survival, that is Dimitri's DM71, shown in maroon over here. And we have also one that actually decreases larvae survival, and that's HPD12-1. That's the sort of teal colored line over here. The rest, however, didn't give us much of a statistical difference between the lines, but yeah, that's 2. We just have this 2 to go on further. So we have only done for the phenarimoles analogs. I haven't looked into the log D just because it hasn't been calculated yet. And I suppose we need one of your help or Ben's help again to get the log D values for that. Then we can look into the sort of relationship between log D values and in vivo significance. Yeah, and we would like input on how to sort of further analyze these the phenarimoles data really, in terms of the compounds and structures and all that.
So on
Can I ask a question, Wilson?
Sorry, on here you've added sorry, I'm just slightly confused about whether these results on the screen here include the compounds that were in our original paper.
No, it was just this batch from 2020.
Okay. So what we want to do is combine those results with these as a complete log D analysis of the whole set.
Yeah, we can do that. Yeah, that's great.
So Ben Perry could be asked to calculate the log D value.
If I'm not mistaken, DM7-1 has a piperidine substituent, so it's totally new. Dimitri was working on other analogs of it, right?
Sure. Yeah.
Yeah, when I checked his ELN, unfortunately he's not here, but maybe Ben can scrape Dimitri's ELN and calculate log D.
So we can easily put together, the all of the molecules have been tested in vivo and just calculate the log Ds, no problem. I guess it would Wilson, if you had that list of every phenomenon analog that you have in vivo data for in a sheet or something, then we could easily just get Ben to put the log Ds in.
All right, I'll put that up on the master sheet on GitHub.
Great. I mean, that's the first correlation that we need, right? Because then the other thing, if we have that that correlation of all of the data that we have so far, then I mean, one thing we should do is take up the offer that was made a while back of access to another small subset of the EpiChem library. And because we can easily just churn out the log D values, the predicted log D values of all of those compounds that are in that library. And there's a large number of things in there, right? And we could then just focus in on compounds which are expected to have very favorable log D values, which we haven't done, I don't think. You know, we could request a sample of 10 or something that just have very low log D values and focus in on those.
Yeah, but maybe we should not only focus on log D, because of course that's our hypothesis, but maybe there are other features about these molecules which makes them different from the others, such as charge or certain side chains, which makes them more similar in other respects.
Yeah, I mean, log D is meant to capture some of the charge out of it. It doesn't capture charge completely, but it is meant to accommodate for that a little bit. So it's not log P. It's meant to be, you know, that's the advantage of using log D, is that it captures that. But I know what you mean. Yeah,
because if there's a special side chain in DM7-1, which is not in the other compounds, maybe that's also one of the reasons.
Yeah, sure. So I mean, with that complete list, we can easily generate the structures alongside the list and see if there's any sort of features that maybe are interesting.
Yeah, because I'm not a chemist, but we, of course, we do have the data of these other, the pandemic, the stasis, and the pathogen box as well. So if a certain group in a molecule makes it important, then we could verify it from these data maybe as well. Although it's a different target, so it makes it very difficult and would be easier to compare in the Fennery moles, of course. And the other aspect is that we now focus on the larval survival, of course, so on the grain. But for me, it's also interesting to know what makes it to penetrate the fungal cell, because for a compound to be effective, it first needs to penetrate the grain and then the fungal cell. And now we're focusing on what makes it to penetrate the fungal grain. But maybe it's also important to know what it makes to penetrate the fungal cell. So from the in vitro data. And if we can combine both of these into chemical rules, for instance, then we might be able to pinpoint what is necessary to penetrate the grain, but what is also necessary to penetrate the fungal cell.
So you mean that the in vitro data tells us about the penetration of the fungal cell?
Yes, because then you take the grain out of the equation, you only have the fungal cell because it's the hyphal fragments, but that's important data as well. So then you get a certain subset of features needed to penetrate the cell, and then you can add the characteristics needed to penetrate the grain, because all of these compounds here were able to penetrate the fungal cell, because otherwise they weren't selected, because they were selected based on their in vitro potency. So they would all have something in similar, which makes them to penetrate the fungal cell, which stands out from all the others. But they have something extra, which allows them also to penetrate the grain, so probably the cement material. It might be good to pinpoint that as well.
Yeah. So I mean, essentially, you want a 3-axis graph, right? You want area under a curve. You want log D maybe, or some other physical feature, and you want in vitro potency. And you want to analyze it with a kind of cluster like that, right? I mean, where ideally the in vitro data if you have a decent larval survival, and your in vitro potency isn't very high, then that implies that it's getting in pretty well to the grain. I mean, I guess that would be quite interesting to, yeah, as you say, those three variables, sorry, those three measurements at the same time. Yeah.
Yeah, and we, of course, we do have a lot of in vitro data, but in the first paper, we didn't address that. So that would be good in this paper to address that as well.
Yeah.
Yep.
Yeah.
So that means that for all the compounds we have, we need certain chemical properties. So log D. So I think we tested by now in total 150 compounds nearly for the Fennery moles.
Wow.
So if we would have the log D of all of those 150 compounds, or some other chemical features like the molecular weight or something like that, we can make comparisons with the in vitro results as well. If we simply plot the IC50 or the MIC50, we do have different values for that in graphs against those features to see if we can find a correlation as well.
So, I mean, in the master list of compounds, then we'll find it very easy just to drop in predicted log D values and obviously molecular weight and so on in that sheet, provided the codes are in there, and provided the however you're measuring the in vivo potency here, so the AUC, for example, and the in vitro data. As long as it's all in there, we can play with the data as much as we want.
Yeah. Yeah.
So yeah, and see if anything pops out.
Yeah, I will prepare the sheet and put it up as soon as I can.
Great.
Yeah, so that was sort of from our side. And just before I forget, Hong, I probably need some molecular weight numbers for a few of your compounds. I just missed it. I'll send you an email. Just, before I forget.
Yeah, no
I will turn off my. Yeah, so that's from our side.
Wonderful. All right, like I said, we've got no chemistry update from Sydney. Sorry, from London this time.
Yeah, so I'll just let me share my screen. So these are just what I've done up to the end of last year. Just give me one moment. So I've done for last year. So these are sort of like my what I wanted to do, but I only finished up to row D, with the exception of one on row E here, which were just tested and were on Wilson's screen from before. So I've done up to here plus that one, and I was not able to complete this because of a shipping delay from Sigma. So basically I was just waiting for these red precursor to arrive, and they haven't yet. So yeah, we're just waiting on that. But given the fact that some of the result that Wilson just shared, I probably deprioritize some of these from this year and probably just do a few more, probably maybe 2 or 3 more of that, and plus that one there down the bottom of row G, and I'll have to think of a couple of more targets for this year as well. Right now, I'm actually focusing on a slightly different path, a different section of my PhD as well. So I'm actually doing finishing up my a review as well. So the synthesis will come after that. So I'll have a review to do to sort of like the end of February. So and then I need to submit an ethics for a different side of my PhD as well. So but I'll come back to do the synthesis, and I'll also send these as well over to for you guys in the Netherlands to test as well. So yes, so that's kind of like quick update of what I'm up to as well.
Which of the compounds on the screen have been evaluated?
I know this one has been, so that was just on screen from before.
What about the one with the spacer, like 53 in row D?
No, this one hasn't been evaluated yet. So I will send some. I've sent a couple of these, but I can't remember because I don't have the list here with me. But this one has not been evaluated yet.
But on row E, you say that 33 there has been evaluated.
Yes. So this one I synthesized like from scratch, and then I was having trouble just replicate that result, so I decided to buy this this bit here. They just haven't arrived yet.
But 33 was inactive.
33 was on like it's good in vitro. Is that right, Wilson? But when I went to in vivo, it didn't have a high level rate.
Yeah. Let me double check again and look at the whole sort of figure. So 33, 3p3. Right, hold on. Do you want me to share it?
Yeah, sure.
Okay, hold on. Is it up?
Yes.
Sorry. Yeah. So 3p3, the pink one over here. Yeah, there's no significant difference, but there seemed to be a slight prolonged larvae survival in the beginning for a little bit. But I mean, we can't tell from much from these lines.
But it was tested in vivo by virtue of the fact that it showed activity in vitro.
Yes.
Okay, all right.
But if I'm not mistaken, the cyclohexyl compound was pretty weak, right?
I mean, I'm just trying to figure. Yeah, I mean, trying to figure out whether it's worth you slogging to make the others in that set you had there.
So that's why I thought, given the result that we just got in, I will need to modify that. So don't do all of them. Just like do something else. I just don't know what something else is because I haven't prepared for it yet, obviously. Yeah. And from the in vivo result that Wilson just shared, so the difference between 12-1, which was the one that's decreased survival, versus 14-1, which was the control from so this guy here and that one. So the only difference is actually 12-1 was the precursor to 14-1. So that 12-1 is 12-1 is basically just 14-1, but with this bit here as a block group instead of this. So that's just a one functional, like one motif change. So I just thought that was interesting to note.
Maybe potency correlates with chain length of the ester of the carbamate functionality. So propylchloroformate, is that a compound you can get, or something? Yeah, propylchloroformate, is that a compound you can get?
I don't have an answer, but I'm not too sure.
I was gonna say, whack another carbon on there and see if the potency goes up, right? Because if the Bach wasn't good, but the ethyl was good, what happened about the propyl?
Interesting.
I think, yeah, I think we need to finalize this complete analysis that Wendy was just talking about to try and figure out if there's a pattern. So I think
really
we'll
don't
make
have
better choices
very good.
when we have a good comparison of the in vivo and in vitro, and then various physicochemical properties.
We really don't have very good SAR on this, correct? Other than knowing, we know a little bit, but not the full picture. Is that right, Dr. Todd?
Yeah, I think so. But at the same time, optimizing things in vitro is really only so useful. You know, we need to have the correlation, which is why these recent results are so useful. We need to have the correlation with in vivo. Otherwise, it's an academic exercise rather than something practical.
Yeah, we need to get something that works in an animal model. Man.
So yeah, okay, but I mean, if these data are going to be added, then that's awesome. Yeah, okay, great.
So I have a
Anyone
little
else?
bit of synthesis too, if you guys want me to do a quick update on that. It's
Great.
not a lot, but it's enough to share.
That'll be good.
Sorry, it's not coming up with my PowerPoint when I'm trying to share. There we go. So basically I was just looking at a couple of the compounds. So there's 3 that I'll show you guys that I've pretty much finished. I've worked through the synthesis and I've got it to work. So I'm just bringing through a little bit more of the starting material so I could do it on a little bit bigger of a scale. So I can do characterization and then also have enough to ship to you guys.
Could I propose a suggestion? I'm sorry, I just saw that you have that ketone down there from slide 5.
Yeah.
Could you react that with O-methyl hydroxylamine? Because we had an oxime series for a little that Dimitri, I guess, just couldn't come. I don't know what happened, but there were no updates from that. Would it be possible to make an O-methyl oxime of that ketone?
I can try.
That's related to this this ketoxium idea, where I think the original compound has another CH2 spacer between the 2 aromatic rings, but yeah, it has that motif in there. The data that Wendy generated a while back had that as one of the actives, particularly when there was a long alkyl chain on the oxygen. It looked pretty promising. So Dimitri's been trying to investigate that. This would be a very similar structure, except without that extra CH2, the extra methylene between the rings.
Yep. Okay. I'll see if I can find some stuff on that and give it a go. Yeah, but basically I've just been making these pyrazine rings through this bis alkylation and then reacting it with the ketone through reductive amination, and this seems to be working fine. So the part that changes is the functionality on the aromatic group here. So we've got the CF3 in the 3 position and then the 4, and there's a nitrile one as well. So having success with those, just got to upscale a little bit. And then I've also been looking at some of the series 2, the 2 amino thiazoles as well. So I'll just share another one of my screens. So I started off with trying to remake the hit target that we had for that one. So basically this is the synthesis route I was looking at. Doesn't mean too much, but basically done this bromination step here, and that's working quite well. And I'm working through to get the other intermediate so that we can then react that and form that final product, and we can retest that to check the activity again. And I've also purchased some of these starting materials with the nitrogen at different positions around the ring and without the nitrogen as well. So we can investigate the position of that and whether it's important, which I think it probably will be, given that that has been important for some of the other compounds we've looked at as well. And some other people that have studied these have also found that without it, they lose a lot of activity. So yeah, I've started.
Right-hand side nitrogen with the methyl group, the methylpyridine.
Sorry, can you just say the start bit again?
The methylpyridine group on the left-hand side of 6357.
Yeah.
Could we try to play around with that too? Because
Yes.
that seems a bit of a liability metabolically, right?
Yeah, so I've only started with the ring structure at the other side for the moment, and I'm playing around with that. And then I'm going to also play around with the other one.
Okay.
We've tested some compounds on GitHub. So once I have that side worked out, then I'll play around with the other side as well.
Yeah, I wish you the best. That's hopefully really good.
This motif, as I understand it, is quite well explored in other areas. I guess this is a core to keep an eye on in terms of what else might be available, through commercial suppliers. I don't know if you've done, searches on what you might be able to buy here or what we might be able to secure from somewhere else.
Yeah, so we did a little bit. There's a table on GitHub with Ben suggested some structures, and then we've gone through and seen where we could purchase them from. So I've just started with some simple ones, and there's a couple that we don't really find anywhere to purchase. So once I get the synthesis route going, I was going to try those ones, and we can buy the other ones or possibly have people donate them or source them from elsewhere. But if the synthesis route works quite well and the reactions all seem to be quite quick and easy, it might not be that time consuming or that costly for us to do it if we've already made the intermediates. So, but yeah, just starting with a few of them and try and get the route working. And if it works, then we'll move on from there.
I was going to say the two nitrogens in that position kind of reminds me of the open source antibiotics series 2 molecules.
Yeah.
So I wonder, almost, if not just for curiosity, could we theoretically send 1 or 2 of those over to Wendy?
Maybe. It's a long shot. I mean, metal chelation possibilities are all over the place, right?
Yeah, I mean, it's really a long shot, but I tend to think in that kind of fashion.
Yeah, it's possible. That's great.
2 more compounds for us to screen will not make that much of a difference, so that's always a possibility.
I think what we can look at is if we go back to this 3 boxes and the phenylmole analogs that we've tested, if these, well, let's say any sort of compounds has that sort of methylpyridine group, if you look into that, maybe we can already know if it works or not in vitro and vivo. That would be a quick shot. Suggestions.
For the other pandemic box compounds, I mean, you've already mentioned the olorifin structure as being really promising. And there were the 2 others, the benzimidazole, and there was another one. And I'm sorry, I forget the structure of those 2. But I mean, those looked pretty good from your point of view. I mean, you're still interested in those compounds? Are they good enough in vivo?
So there's ravuconazole and azole. There is a benzimidazole called fenbendazole.
Yeah.
And then there's an MMV compound, 173837 or something along the lines. I think the MMV compound would be something to look into in terms of the structure and all that. Well, we already know that azoles work for the fungi, so I mean, I don't know if we need another azole.
No.
Right.
Benzimidazole.
Seems like olorifin was the one that was the sort of surprise.
Yeah.
Is that right?
Well, not really. It worked really well. Yeah, I mean, we've done in vitro tests last year, and we already know that the in vitro concentration was really low, and we know that sort of it may work, and it didn't come to a surprise. What came to a surprise was that the compounds that we think may increase larvae survival did not. For example, tafenoquine, that's another one. So a compound called tafenoquine is, which is quite actually. Let me just share my presentation again.
Yeah, that's got a very interesting molecular structure to it.
Which one?
Tafenoquine. It's got some interesting things in it. It's got 2 trifluoromethoxy groups, if I'm not mistaken. So it's very lipophilic.
And the good thing about this molecule was that it was not
Yeah.
only active against Madurella, mycetomatis, but also against other causative agents of eumycetoma.
So this sort of dark green one, smaller circles, it also sort of increased survival. It looks like it's quite similar to olorofim, the lines, but it just sort of, it dips a bit towards after day 7. That's why there was no significant difference. But if you sort of look at the line, just the lines before day 7, it looked like it would. I mean,
but
this
we
could
also,
do with, yeah, sorry, Wendy, you go ahead.
yeah, so what we need to take into consideration is that we can only treat the larvae in the first 3 days. So that means that after day 3, they don't get any injection. So I don't know what the half-life of these drugs is in the larvae. So that could also be some effect there.
Yeah. And also we've only done one drug concentration throughout our in vivo studies, and we infect them, we treat them in day 1, day 2, day 3. Maybe, I mean, if we play with this concentration and the infection times, treatment times, the graph, the figure would come out different. So there are also some things that we actually still not sure. But tafenoquine is something interesting to look into, because it not only inhibits Madurella mycetomatis, it also inhibits 4 other sort of very close, 4 other common mycetoma causative agents. So yeah, I mean,
even the one resistant to most of the drugs, because we tested one of them, Metacopsis romeroi, and it's resistant against almost everything. But tafenoquine was able to inhibit it, at least in vitro.
Did you test P. boydii as well? That's another notorious drug-resistant bug.
no.
It's very interesting. I mean, I guess we're also interested in novel mechanisms of action here, right? Which is one of the reasons why olorofim was interesting. That's the DHODH inhibitor, is that right?
Yes.
Yes.
Okay, which is interesting because it just gets into a new place. And I guess I'd be interested to know what the other MFV compound, the 387 compound, what that does, if it's known, just in case that's interesting too.
I think it may be known. I'll open up an issue on that. I remember seeing something about it somewhere. I got to go through my own files.
I mean, from the curves there, I guess the fact about you can't treat it after day 3 is interesting, because I mean, obviously none of the compounds are breakthrough above 50% survival after 10 days, right?
Yeah,
they are still going down, but you're saying that because you're not able to treat after day 3, that maybe it could have been better if you could have maintained treatment. Is that what you mean by that?
Yeah, that could be one of the reasons, but yeah, we simply can't, because it causes too much injury to these larvae, and they die due to the injury, not to the infection.
I see.
Yep.
So actually, the first 3 days are some of the most interesting then.
I say usually with fungal infections, the quicker you get to them, the better the outcomes are.
Yeah.
Yeah.
That's true.
But I agree with Ben that olorofim is certainly a drug which is very interesting. The only thing is, I don't know, I didn't look into the patent of olorofim, for instance, if we are allowed to play around a little bit surrounding this target. And if there's something we can learn from its structure. Of course, it has a different mode of action. So combination of olorofim with an azole would also be possible, maybe. But I also agree with Wilson that tafenoquine might be interesting as well, to see if we can get this activity to last all the way, or make it a little bit more promising if we play around with this compound as well. Yeah, I'm open to both.
Yeah, I also just had an idea. Since olorofim hits, folate DNA synthesis sort of thing, have we tried daraprim in an azole?
Sorry, I didn't get that one. Which one?
Daraprim, you know, the drug that was whipped up in a lot of controversy back in 2015 for the price hikes. It's a folate inhibitor as well. Similar pathway as olorifim.
No, we
That's an
didn't
old-
test that.
Yeah, Matt, isn't that's a folate inhibitor, right? Daraprim?
I can't remember. Sorry, I can't remember. It's a toxoplasmosis compound, right, which is also used for the treatment of malaria. I
Yeah,
can't remember what the mechanism of action is.
it's full. I'm sorry. Let me get Wikipedia. I'm trained to be a farmer. I'm gonna be going to school for a pharmacy tech anyway, so probably gonna end up having to reference a lot of these things. Yeah, it's an anti-folate DNA synthesis inhibitor. Maybe combine. Maybe we can try it.
Certainly inexpensive to make.
Yeah, dihydrofolate reductase. That's the target. DHFR.
Okay. Yeah, it's an interesting question about freedom to operate around olorifim. I don't know. I would hate to try and answer that question. I think one is free to do basic research now, but it depends how far you take it. I don't know at what moment you begin to impinge on someone's activity. I don't know the status of the drug, actually.
Yeah, also looking at these graphs, I wondered for the azoles, we have tested in the last years a lot of azoles as well. And a few of them had activity at this concentration while others had not. Since the azoles have the same target as the Fennery moles, again, I'm not a chemist, but if we find a certain chemical rule, would it help us also to verify if that same rule applies to a different class of antifungal agents, but active on the same target?
The
Then we
azole
the
group
data.
in our drug, the azole group in the drugs, and the pyridyl group in our phenaramoles basically work to bind the iron center of CYP51. So they're basically very similar drugs, even though they look different.
I think if you maintain the same mechanism of action, it's likely that you can make a better comparison. Sure, I would suggest.
Yeah, so maybe it's worthwhile to make the comparisons in the Fennery mole first and then verify if it's true for the azoles as well.
Yeah, sure. Yeah.
Or the other way around.
Yeah.
Mm-hmm.
It's best to look at these as these. It's best to look at the. Sorry about my voice. Phenaramoles as a sister class to the azoles.
I mean, again, it's a relatively simple calculation, right? So if we cluster together in the master list some of these compounds that have been evaluated, I mean, they can just be put in the master list themselves. It's no problem, and then we add in the smiles, and we get the log.
There's probably scripts to do this already.
We can easily extend the comparison, no problem, and that should be reasonably meaningful, I would have thought.
Yeah, because the azoles, of course, are still the drug class used to treat mycetoma. So if that could make some suggestion which compounds are more likely to be active and which compounds are less likely to be active, that might even mean something in the clinical situation already. I know it's far-fetched and it's far away, but yeah.
Wendy, what's the azole of choice?
Currently, it's itraconazole, and raviconazole or fosraviconazole is the one in the clinical trial from DNDi now.
Just coming back to the thing you mentioned about combinations. I mean, obviously, all of these experiments are single molecules. Do you ever look at combining two molecules that look promising in these kinds of experiments, or is there something difficult about doing that, or?
Well, in the past, we did combine, for instance, an azole with terbinafine or an azole with amphotericin B. That was before we had all these data, of course. And then with amphotericin B, we had enhanced survival. But when we combine it with an azole, we see actually some antagonistic activity, because the promising activity of the azole was lost. Same when you combine it with terbinafine, but of course that's targeting the same pathway by different classes of antifungal agents. So it would be more promising to combine, for instance, an azole with olorafim, although in vitro we didn't find any synergy at all. We also didn't find antagonism, but in vivo we didn't test it.
Right. Right. Interesting.
Because usually fungal infections usually are treated with a combination of drugs.
Yeah.
Well, the majority of the fungal infections, I think, is treated monotherapy only.
Well,
in the Netherlands, we treat aspergillosis with a combination because we have a relatively high resistant rate in azole-resistant and aspergillus. But the
I was
rest of
thinking
the world,
more along the lines of the more severe stuff, like cerebral phaeohypomycosis is treated with a combination of drugs, for example.
yeah, but that's because of the blood-brain barrier, isn't it?
I don't know though. I mean, so much still unknown, right, about fungi.
I mean, it doesn't really help us with this, because
No.
of course we're trying to find a molecule that's potent. But I mean, just in terms of, you know, if you had to take a guess at a way of improving some of these survival curves, then a combination could be a way to go. But at the same time, that may be us losing focus on the need to find a molecule which is potent on its own and has an interesting mechanism of action and does things that we don't currently have. So it's just more a question about whether that's ever included in the research workflow. Very interesting though. Okay, well, great data. Thank you. All right, was there anything else? Just because we're coming up to the hour. I was just going to mention three things, which I've mentioned on the agenda. One is we tried recently to get a grant funded on some of this work and got knocked back. So we're still thinking about ways of putting in applications for, you know, a significant amount of research funds to get things going. I mean, that's kind of bubbling away, and Wendy and I are talking, and we're talking with Ben and things like that about ways to get some funds for this to keep things operational. So no positive news to report yet, but we're still working on that. Second thing was just the work that Dimitri and I think Hung were doing on the numbering system for the molecules. So all of the molecules that are now in the master list have been assigned a unique number based on our numbering system. And if we can, it would be great to use those, just so that we know exactly which molecules we're talking about and everyone's using the same code. That would be really, really great. So there's a new line there's a new column in the master list which has those. The way it works is you have a number for the molecule, and then you have a batch code and you have a salt code. And for discussing molecules, you don't need that extra stuff for the batch and the salt form. So it's just really the number for, for slides and stuff. We just need the number of the molecule, but it just means we're never going to get confused about which molecule we're talking about. And also, if we are talking about two batches of the same thing, we've got that covered because we can assign those codes to those batches.
Yep.
And then the last thing I was just going to mention very briefly is that on Saturday we have World NTD Day, and I put up an issue about the starting off with a newsletter about the project, which we could send around. So the idea is that we write a newsletter on GitHub, which works really well because you can put links and photos and stuff, and it's very easy to copy that into an email and send it out to people. We've done this with the antibiotics project, and it works really well. And people like getting emails, email newsletters, because it's an easy way of updating everybody. So the advantage of doing it this way is, of course, a newsletter is on GitHub, so anyone can read it, but you can also email it out. So if people don't get it by email, they can still read it. To send things to people, you need their approval, their permission to do that, which means people have to sign up to it. And we've done one of these sign-up lists for the antibiotics project, and it's very easy. You just use your Google account and do a Google Sheet, and of course people put in their details and it goes into a spreadsheet, and you can copy the emails and send it to everybody. but we do need to do that. So there is one, I put an issue up on this, and Dana Klug on the antibiotics project made one. I was just looking for someone who might want to do that for this project, using the open source Mycetoma Gmail address to set up a sign up page, which is really, really simple. But I haven't had the bandwidth to do it. If no one wants to do it, I mean, it's a big part of the outreach, right? So to engage the public in what we're doing. If no one wants to do that, then Dana said she'll do it, but it just needs to be done before Saturday. That's all. So there's an issue
head off,
there.
everybody. Just want to wish you all the best.
Thanks.
See you again on Friday, Matt.
All right. There's issue number 42 and 43. If people could take a look at that, that'd be very helpful. If no one wants to do it, I'm going to have to ask Dana to do it, because I think it's important we try and do something on World NTD Day, because it's a massive outreach opportunity to highlight things that we're doing, and we should join in that big thing, because there'll be a huge thing on Twitter with a hashtag and everything, and we can highlight what we're doing and maybe get people to look at the project. So there's a draft newsletter. People please suggest things that we can put in there, whilst keeping it short and sweet. And we need someone to do the newsletter sign up. That's the crucial thing, ASAP, so that we can put in the newsletter on GitHub. If you want to receive newsletters, go here, because we're not allowed to send it to people who haven't agreed.
Maybe we should Twitter it as well.
Oh yeah, absolutely. So what you can do is you can tweet it out and everything, and have a link to the newsletter, which is on GitHub. And if people want to receive them in the future, they can just sign up.
Yeah.
Maybe Wilson and Ma, are you able to make such a list? Ma is, I think she is, her camera is off, but she's our new PhD student who will be joining in this project as well.
Hi.
Yeah, I think, yeah, I mean, Maya and I can sort of look into it. I'm still a bit lost on how to actually start doing it.
I can do it if you like. I've done things like this before, so.
Oh, great. I mean, Hung has the sign-in for the Gmail address.
Yeah, okay.
And so you just need to log into that account and use that to create the sheet and the form, that's all. And
Yeah.
that's helpful, because then none of us have to worry about maintaining the email address, right? Because we already have one.
Yeah, all right, I'll put my name on that then.
Great, cool, that'd be great. So then when we tweet this out on Saturday, we can include that. Yeah, awesome. If you have any trouble, just contact Dana, because she did it before. Awesome, great, that's everything I have.
Yep, on my side as well. I'm not sure if Wendy wants to add anything.
No, as Matt mentioned, we're looking for funding. There are maybe some opportunities with the European Union. I will look into that further with one of our co-workers who had such a grant to see what we actually need to be able to apply for that. Yeah. And for now, we're hoping to finalize the last data on the Fennery Mole paper so that we can put one updated paper out there as well. I think that would also be good to keep the momentum, to keep people interested in this, also from outside this small community here, just to demonstrate that we're still working on this and we're trying to get some answers out of these data.
All right, great. Nice to see everybody.
Yep, thank you. Yeah.
And are we sure about the next meeting time?
Oh, I think the calendar for next meeting, I think it ends in December. So that's actually, when I checked it, there's no next entry for next month. But like the following month, when it's this timetable is on, I think we might have to fix time invite again. Yeah.
Okay.
So I think, can you guys go and check? Like I don't think, because when I check my calendar next month, there's nothing. So one of the invites was not working because it ends last year essentially.
I have it in my calendar.
For when?
Tuesday, the 23rd of February at 11 p.m. Sydney time.
I've got nothing.
So it's
Okay. Shall I try the ICS file again?
Because when I opened it, it say the end date was 31st of December 2020 for time A.
Is that the newest one that we made or the old one? There's a
In my agenda, it's also displayed.
Because
I think
Kimberly,
so.
you posted something a couple of weeks ago.
Yeah, so that's the one I have running on my Outlook and it seems fine, but maybe we can
That
just,
works for me as well.
yeah.
Okay, I'll try that first.
So I think it's 1 o'clock our time, Mandy, over here on 23rd of February, if mine is correct.
Yes, it's 1 o'clock Amsterdam time.
Yeah, 23rd February.
It's very civilized.
Yeah, so I basically just extended the time to like the end of this year. So I do actually have it on the 23rd as well in February. So is that the fix is easy? I'm just not sure. Like, is it just me that has the problem, or is it someone else found it as well?
I'll try the new ICS file that Kimberly posted and see how it goes.
I will put the figures on GitHub in the PowerPoint, and then I'll put up the sheet as soon as possible. Most likely by the end of the week. Yeah.
It's Wilson. Is it also possible if you can rearrange the graphical data for some of my compounds? Because I think we might be able to tease out a little bit more about the relationship between the structure and the
Yeah, sure, no problem.
Yeah. I'll post a because they kind of come into two sets. So I'll show them what the structure looks like. Maybe you can regroup them into the two graphs slightly different.
Okay, yeah, just sort of tell me what to do, I'll do it.
Yeah, thanks.
Yeah, and maybe we should make the new numbers in the graph. Because now they are now displaying their original numbers, of course, but yeah, since we're now having this system, let's use it as well.
Yeah.
Great, no problem in putting the little ones, the old ones in brackets, right? If it helps in the in the painful transitional period, but yeah.
I'll do that. I'll make a little bracket.
Matt, and also in like going forward in the future, who will be in charge of actually updating that that unified sample code when new one comes in?
Oh well, I mean, if you make a molecule, then it should just go in there.
Okay.
That's part of the workflow of synthesis, and also if I guess we've never had a unified thing about this, but to my mind, if a molecule is proposed and then is synthesis undertaken, we can at that point put the molecule in the sheet too. But in that case, we'd need another column which has an indicator about whether a molecule has been completed or not. So it's a very simple way of keeping track of suggestions.
Yeah.
But we just need to have an easy way of filtering them out if we don't want them there. That's all. But there's no problem in assigning a code to a molecule because there's an infinite number of codes, right? So yeah, if something is strategically important, we can stick it in there and say proposed or something like that. But certainly when it's been synthesized and certainly when it's been evaluated, it should be in there with a unique code. And you take the code that is next. So you can sort the sheet based on the codes and you can see the one that's next, right? It's very important if the molecule is in there already that you give it the same code and you change the batch number. That's also really crucial. So
Yes.
you don't accidentally give it another code, because you've forgotten that it's in there already. So a quick search on the SMILE string is important.
Yeah. So basically, if we proposed a compound or some compounds and someone started to synthesize it, then that should go into the
I think it helps.
with a unified code.
Yeah, I think it helps because then we can talk about it more easily. And there's a link there to what it is. But in that case, if we do that, which we haven't done so far, we need to just insert a new column and you put a 1 against things that have been made and a 0 against things which haven't. And that easily allows you to digest the spreadsheet.
Yeah.
Not get confused. Yeah. Okay, so yeah, it works better if it's the person who originates the molecule puts it in.
Okay, no problems. I'll start doing that when I start synthesizing again.
Great. All right, great. Nice to see everyone.
Thank you. Have a good
See you
evening
next
or
time.
afternoon or day.
Take care.
Bye, everybody.
Yeah.